Yeah, I can. Okay, so today we'll be talking about Campbell chapters 5 to 7. So Campbell chapter 5 is the structure and function of large biological molecules. So what are macromolecules? So macromolecules are polymers made from short monomer units. So mono stands for one basically, and so it's like a one small unit of I guess like a polymer then. Polymer poly stands for many, so it's synthesized from many short polymer units. So monomers are synthesized from dehydration reactions, which are anabolic, and then that involves taking an H from one side and an OH from the other side, and then taking out the water, and then you have a longer polymer. It's also broken down by hydrolysis, where you add the water molecule in between the units you want to separate. So monomer chains, as you can see, end in hydrogen or hydroxide, and then the two are taken out or added back in. So first we'll be talking about carbohydrates, which are sugars and polymers of sugar. So the monomer is a monosaccharide, and the chemical formula is some multiple of CH2O. So for example, we have glucose, which is really common, and then its chemical formula is C6H12O6, which is in the CH2O formula. So carbohydrates have the carbonyl group, which is a C double bonded O, and also a hydroxyl group, which is OH. And its polymer, if it's two monosaccharides binded together, it's called a disaccharide. And then more than two monosaccharides is called a polysaccharide. So the monosaccharides are joined by a glycosidic linkage. And there are two types of glycosidic. Well, there are more than two types, but the main two types are 1,4 and 1,6 glycosidic linkages. So for a 1,4 linkage, as you can see on the bottom green monosaccharide, the 1,4 linkage is basically formed by having a 1 carbon, as you can see notated by the numbers, binded to a 4 carbon. So that forms a straight line of monosaccharide units. Whereas the 1,6 glycosidic linkage is formed by taking a 1 carbon and then binding it onto a 6 carbon. So that creates a branched polysaccharide structure. So similarly, there are two types of rings. There is the alpha ring, which has the OH groups in the same orientation, and then the beta groups, which have OHs in opposite directions. So these two structures are interconvertible, meaning that they're like interchangeable. Well, they exist in like basically the same ratios. And so polysaccharides have the polysaccharides' main functions include storage of energy in the forms of starch and glycogen, and also structure in the forms of cellulose in plant cell walls and chitin in fungus or exoskeletons. So carbohydrates are named after the number of carbons in the carbohydrate in the oh my gosh, in the monosaccharide chain. So for example, if you have 3 carbons, it's called a triose. If you have 5 carbons, it's called a pentose. And if you have 6, it's called a hexose. So here, this diagram is also showing how glycosidic linkages form. So the OH and the H are taken out in the form of a water molecule, and then the two monosaccharides are joined together. So next we'll be talking about lipids, which it's also commonly known as fats. So its main characteristic is that it is very hydrophobic, since, as you can see in the top right, the diagram, the lipids don't have any OH groups on the actual fatty acid chains itself, which is and it doesn't have any other polar functional groups either. And CH is always and CH is very nonpolar. So fats are composed of a glycerol group on the left, which is in the gray, and also fatty acids, which are in the yellow. A fatty acid is a 16 to 18 carbon skeleton, and it could vary in number. So 3 fatty acids and a glycerol group form a triacylglycerol, which is pretty self-evident by the name. There are 2 types of lipids, saturated lipids and unsaturated lipids. So saturated lipids have saturated fatty acids, which do not have any double bonds in their fatty acid carbon chain, meaning that this chain basically ends up being straight and there are no bends in the tail. And then unsaturated fatty acids have double bonds in the tail, and then that creates some bends in the tail. So saturated fats are usually solid at room temperature and are unable to move a lot because they lack the bend in the CH chain, which ideally creates more space for them to move. Whereas unsaturated fats take a more space because of their bent tails, and then that results in liquid fats at room temperature. So an example of a saturated fat would be butter or animal fat, and then an example of unsaturated fats are vegetable oil or olive oil. Okay, wait, let me answer. Can you explain the differences in chemical properties that are caused by the 1,4 and 1,6 glucose chains? So go back like 2 slides. Okay, so the 1,4 linkage is where, okay, hold on. So 1,4 is created by having a bond on the one carbon and then binding it to the 4 carbon. So that basically creates like, I don't know, like a longer singular line chain of monosaccharides. And then a 1,6 linkage basically links this 1 carbon to the 6 carbon, which basically creates branches. So okay, hold on. And then the chemical properties, they don't really change, but the function kind of changes. So when it's more branched, I guess they function more in structure. Okay, let me clear everything. Okay, I'm gonna continue first. Can you like go back to the
What?
Yeah, okay. And then lipids can also form steroids, which contain 4 fused CH rings. So an example of steroids are cholesterol, which can be found in the phospholipid membrane. And then steroids also can function in hormone signaling and other cell signaling. Then next, I'll be talking about proteins. So proteins is a complex of many polypeptide polymers in a specific 3-dimension structure. So here is the general structure for an amino acid, which is a protein's monomer. So there is an amino group, which is NH2, an alpha carbon in the middle, a carboxyl group, and a side chain, also known as an R group, which can vary by amino acid. So every amino acid has a different side chain that sets it apart from other amino acids. So the polymer is the polypeptide, and that is formed by linking amino acids with a peptide bond. And protein functions include being an enzyme, being a hormone, participating in defense with like antibody proteins, transport proteins, structure, motion, receptor, and storage. So here are the side chains that I was talking about. So there are hydrophobic side chains, which obviously are hydrophobic and they don't like water, as we previously talked about. Okay, next.
Oh, memorize, by the way.
Yeah, oh yeah, you have to memorize all of them. And then there's hydrophilic side chains, which prefer to be with water. Can you go back?
Oh, you can search
Okay.
this up, but you memorize it.
Yeah. And then it's all in Campbell's too, so just go in the book. And then there's also acidic, which are negatively charged, and also basic, which are positively charged side groups.
Go back where?
Check Campbell's. This is from
Check
Campbell's.
Campbell's. Yeah, Campbell's online. You see, remember like which ones are non-polar, which ones are polar,
Yeah.
and then which ones are
Test,
charged,
they
and
test
then
like
like what charge they
specifically
have.
the amino acids.
I mean, they only test like polar and non-polar interactions.
They
So
also had like specific names. You just need to, yeah, you just need to know which ones are polar and which ones are non-polar. Okay, so next, there are 4 levels of protein structure that these amino acids can form. So the primary level, the first one is just amino acids linked by peptide bonds, as you can see here. There's like a huge chain of oh yeah, Anthony, can you like answer chat questions? Because I can't always see them. Okay, so the primary structure is just a chain of amino acids. And then the secondary level, it's formed by hydrogen bonding interactions in the polypeptide backbones, which folds the protein into alpha helixes or beta sheets, depending on the structure. So these are not interactions between R groups. These are interactions between the oxygen and the hydroxyl group, which is the C double bonded O and OH, which has the oxygen has a slightly negative charge, and so those interact with nitrogens attached to the those interact with hydrogens which are attached to the nitrogen and have a partially positive charge. So next, the tertiary structure is determined by interactions between side chains or the R groups, as we talked about. So amino acids with hydrophilic side chains tend to stay on the outside of the protein since the cellular environment is very water-based. Meanwhile, the amino acids with hydrophobic side chains tend to stay on the inside of the polypeptide and away from the water. So this is also influenced by interactions between positive and negative side chains, which means that negative and positive charged side chains basically like to stick together. And so that helps hold the protein structures. And then also another specific type of interaction, I guess, is a disulfide bond, which is formed between cysteines, which have sulfurs. So that's pretty important to know. These are called disulfide bridges. And then a quaternary structure is basically an aggregation of multiple tertiary structures. So technically the most basic functions emerge at the tertiary level, but some examples of quaternary structures include hemoglobin, which is made of alpha and beta subunits that contain heme, which is an iron that binds to oxygen, and also collagen. So changes in primary structure can also change protein structures later on, because if you have different amino acids with completely different side groups, then their interactions will be different, and then your protein folding will change. So proteins can be denatured when exposed to extreme pH, temperature, or salinity changes. So basically unfolds and loses its properties. Remember that proteins beyond the primary structure are held only by hydrogen bonding or other intermolecular forces. So they aren't very permanent. So an example would be if you transfer a protein from a polar to a nonpolar environment, the amino acids might reverse themselves, reverse their orientations, because your hydrophilic amino acids, which are originally on the outside, would want to be on the inside, so everything will flip and then everything will change dramatically. So another thing is that chaperonins help with protein folding. So chaperonins are basically little protein capsules that polypeptides enter, and they provide an ideal environment for the protein to fold better.
actually, there's a question.
Hmm.
Is protein folding bad?
Is protein folding bad? No, it's good. It helps your
Why?
proteins. Yeah. It's normal. I wouldn't say like bad. It's just what happens. Okay. Next is nucleic acids. So the two nucleic acids that I will be talking about right now are DNA, which is deoxyribose nucleic acid, and RNA, which is ribonucleic acid. So the difference between RNA and DNA are that RNA is single-stranded while DNA is double-stranded, usually. And also RNA contains ribose while DNA contains deoxyribose and runs anti-parallel to each other. So the purpose of nucleic acids are to store and transport genetic information, and its monomers are nucleic acids. Well, yes. So nucleic acids are formed with a 5-carbon sugar, which is a pentose, and also a nitrogenous base, which is guanine, adenine, thymine, and cytosine for DNA, and then in RNA, the thymine is replaced by uracil, and also a phosphate group, as you can see on the top right. And then there are 2 types of nitrogenous bases. One is pyrimidines, which are single-ringed, and those include cysteine and thymine, and also uracil. And then purines are double-ringed, which includes adenine and guanine. So a pyrimidine always pairs with a purine, and that ensures that DNA is always paired at a specific width. So a little bit about genomics and proteomics. Like, these aren't tested very frequently, I guess. Genomics is basically using sequences of genomes to analyze them, and then proteomics is basically the analysis of large sets of proteins and also their sequences. This is done by bioinformatics, which is using machine software to analyze and process large amounts of data. Okay, the next chapter is, actually, let me see if there are any questions. Oh, Anthony is answering. Thank you. Now, pyrimidines are single rings. Well, purines are 2 rings. Like, hold on. Yeah. Purines are larger because they have 2 rings. And then pyrimidines have 1 ring. Yeah. Yeah, the gluing thing. Okay, I'm gonna move on. Okay, the next chapter is chapter 6, which is a tour of the cell, where I talk about the organelles and we have fun. Okay. But first let's talk about microscopes, because I've seen this get tested. So the light microscope is like very common. It passes visible light through the specimen, which is refracted and then magnified to reach the eye. Microscopes have a certain magnification, which is how much the specimen is enlarged, the resolution, which is how clear the image is, and also the contrast, which is the difference between light and dark areas of the image. Electron microscopes focus beams of electrons onto the specimen. So the resolution is inversely related to wavelength, and electrons have a very, very tiny wavelength, which means that the resolution will be very, very clear with an electron microscope. And so a similar thing is the scanning electron microscope, which what did I write? Oh, but basically you cover the specimen with a thin film of gold. And so the electron beam excites secondary electrons on the gold. And then the secondary electrons get detected. So you get a really nice 3D image. How detailed do we need to know this? So I know that the question I saw was talking about specific techniques used with microscopes. So I guess a little bit more specific than what I'm talking about right now. Doesn't matter which edition. Yeah, 11 or 12 is the best. I've also used 9 before. Yeah, they might be a bit different. I don't remember. Oh. Okay. Anyways, the transmission electron microscope is when specimens are stained with heavy metals and electrons are focused through the thin portions of the specimen. So the electrons will be attracted to the heavy metals and then show basically what the specimen looks like. Hello. Can I have the next slide, please?
I don't know what happened. I pressed A and it changed presenter views.
Oh, okay. So another technique.
I got this. I got this.
Can you?
I
Okay.
got this. I got this. Wait, which one was it?
That one, yeah. That. Thank you. So another technique that is used a lot is cell fractionism. So basically, you take cells apart and then you separate the major organelles by centrifuging. So the organelles and cell parts have different sizes and masses. So usually at low speeds, you centrifuge it, which is like spinning a tube at really high speeds. And then since there's a centrifugal force, the organelles will settle at the bottom and form a pellet. Depending, and they will form different layers depending on the mass and depending on what you really want to isolate. So here we are going to talk about the differences between prokaryote and eukaryotes. So they have similarities, which include the cytosol, which is all the fluid and stuff inside the cells. They both have so actually to define, prokaryotes are like the more ancient variation of cells. So they were the first cells that existed, and then eukaryotic cells, they came later than the prokaryotes. And so both of these cells have ribosomes, so they make proteins. They both have genetic material, which in the form of chromosomes, and then they both have cell membranes, and plasma membrane is the same thing. So the difference is, though, prokaryotes do not have membrane-bound organelles, and then prokaryotes also have a nucleoid, which is a mass of genetic material instead of a nucleus like eukaryotes have. So we all have eukaryotic cells because all animals have eukaryotic cells, and yeah. So also the reason why cells are always really small is that it increases surface area to volume ratio, so that allows for more material exchange. So as you can see on the right, the total volume between these squares remain constant, but then your surface area increases by a lot since you have a little square and then that has 6 sides, and that increases the surface area a lot. So here's a list of all the organelles in the eukaryotic cell. So the nucleus, it stores genetic material, and it's the bright orange sphere in the center. And then the rough ER, it has ribosomes on it, which produces proteins. The smooth ER is in charge of lipid production and detoxification. The Golgi apparatus packages materials and vesicles for transport. The lysosome digests particles, both intra and extracellular. Ribosomes obviously produce proteins. The nucleolus produces ribosomes from ribosomal RNA. The mitochondria produces ATP for the cell as energy. And the peroxisome it takes charge of metabolism and produces hydrogen peroxide as a product. And then there are even more. So the centrosome is the location of microtubule initiation. The cytoskeleton includes microfilaments, microtubules, and intermediate filaments, and that reinforces cell structure. The plasma membrane separates contents of cells from the outside and also regulates material exchange. The cell wall, which is not in animal cells, is a rigid outer layer that basically provides more structure to the cell and also protection. So those are in plant cells and fungal cells. And plasmodesmata, which are in plant cells only, is basically a tunnel that connects the two cells. So I'm gonna go into more depth about the nucleus and ribosomes. So the nucleus is surrounded by a nuclear envelope, which is a double membrane. So the envelope has many small pores to allow entry or exit of proteins and RNAs, which is called the pore complex. And the nuclear lamina lines the nuclear side of the nuclear envelope. So DNA is organized into chromosomes, and DNA wrapped around many histones is called chromatin, and it condenses before cell division. Ribosomes are synthesized in the nucleolus from ribosomal RNA, also known as rRNA, and here is a picture of a ribosome. It has a large subunit and a small subunit, and when undergoing translation, the mRNA goes in between them, and then the protein comes out the top. rRNA exits through nuclear pores to the cytoplasm, where it assembles into ribosomes. And ribosomes there are free and bound ribosomes, which have slightly different functions. So free ribosomes usually synthesize proteins for use within the cell, while bound ribosomes are attached to the endoplasmic reticulum and synthesize proteins for insertion into membranes with vesicles, so that includes into other organelles or out of the cell. It can that is it. So next I'm going to talk about the endomembrane system. So the smooth ER helps detoxify drugs and synthesize lipids, which includes oils, phospholipids, and hormones. So cells that have to take care of a lot of toxins, such as liver cells, have a lot of smooth ER because that is what they do. Rough ER has ribosomes that produce proteins for extracellular use, and they also can produce more membrane for the endomembrane system to grow. So most secreted proteins from the ER are glycoproteins, which are proteins with carbohydrates binded, and these are transported by vesicles to the cell membrane. The Golgi apparatus receives and transports substances. So it has two sides. One is the cis side and one is the trans side. So the cis side usually faces the ER for receiving of proteins, and the trans face usually faces away. And vesicles delivering content usually merges with the membrane, and then vesicles that are going away from the Golgi pinch off from the Golgi. Also, the Golgi slightly modifies products that enter it before delivering. And then lysosomes digest intra or extracellular macromolecules, which include waste or damaged organelles, and this is called autophagy. Lysosomes have digestive enzymes, so a large scale breakage of lysosomes can result in apoptosis or cell death. And then vacuoles can transport solutes or store substances. So for example, cells have food vacuoles that basically store nutrients, and also certain cells have contractile vacuoles that basically remove extra water that come into the cell so that the cell doesn't explode. And then plants have large central vacuoles for storing water. In case there's a water shortage, the cells will still have enough water from the central vacuole. The next are the mitochondria and chloroplasts. So mitochondria and chloroplasts change energy from one form to another. Mitochondria use sugars, and chloroplasts use the energy inputted from light to create sugars. So they came from the endosymbiont theory, which says that basically mitochondria and chloroplasts could have been small prokaryotes that were engulfed by bigger cells. And this proof comes from them having independent genetic material and also double membranes. So the mitochondria has a smooth outer membrane and folded inner membrane, which are called cristae. This gives it a lot of surface area for its processes to be carried out. And the mitochondria matrix is enclosed by the inner membrane, and it consists of a lot of enzymes and other proteins for cellular respiration. Chloroplasts are in plant cells, and it basically, as I said, takes the energy from light and uses it to synthesize sugars. So the chloroplast has stacks of thylakoids inside of its membrane, and each stack is called a granum. The fluid outside of the thylakoids is called the stroma, and reactions take place in the thylakoid membrane and also the stroma. So basically, there's an intermembrane space, the stroma, and the thylakoid space inside of the chloroplast. And then here I will also be discussing a little bit about peroxisomes. So peroxisome basically remove hydrogens from metabolized products and transfer them to oxygen, which creates H2O2, which is hydrogen peroxide. So hydrogen peroxide is toxic, but peroxisomes have compartmentalization and also enzymes converting H2O2 to water to prevent the toxicity. And then hold on. And then lastly is cytoskeleton, which its main function is to provide mechanical support for the cell. And it also has a role in cell motility when it interacts with motor protons. And as you can see, the vesicles are basically brought by motor proteins along microtubules of the cytoskeleton. So it basically walks itself along the cytoskeleton to bring to bring materials. The cytoskeleton is made of microfilaments, intermediate filaments, and microtubules. So microfilaments are the smallest, and then microtubules are the biggest, while intermediate filaments are right in the middle. So microtubules grow out from centrosomes and help with motility. Intermediate filaments make up the permanent framework of the cell and also can reinforce organelle positioning. So these intermediate filaments will actually remain after cell death, so it maintains cell structure still. And microfilaments, also known as actin filaments, bear weight and pull forces. So actin filaments and another type of filament called myosin collaborate in contractions, and this is seen in muscle cells. And it also forms the semi-solid cortex, which is the outer cytoplasmic layer of the cell. Oh, and also microtubules are seen in cilia and flagella to help with motility. Oh, so here I talk a little bit more about cilia and flagella. So they are used for locomotion or to move fluid. So they move fluid in oviducts, for example, to basically move the egg during ovulation. And it's also used in the windpipe to move out debris. And it's used and it's also for locomotion, like on sperm. So they are anchored by the sorry. There are usually many cilia per cell and only usually 1 to 2 flagella per cell. And it's basically a group of microtubules sheathed in an extension of the plasma membrane. It's anchored by a basal body, which is really similar to a centriole, and it is 9 triplets of microtubules arranged in a ring. It is also bent by dyneins, which are motor proteins, and in this case, they are the red little Y-shaped proteins here. And they walk along adjacent motor proteins and then use ATP for energy. So actually, interestingly, basal bodies are very similar to centrioles, and a sperm's basal body, once it enters the egg, actually becomes the centriole of the fertilized zygote. Here are also some extracellular components. So the plant's cell wall protects the plant cell, maintains the shape, and prevents excessive uptake of water. It's mostly made of cellulose, and so first the primary cell wall forms, and then the secondary cell wall forms inside for extra strength. And plasmodesmata allow for material exchange, which are little tubes, little channels between cells. And then the animals have an extracellular matrix, also known as the ECM. So that includes glycoproteins and other carbohydrate-containing proteins. The glycoprotein most abundant in animals is collagen, and I'm sure you've heard of that somewhere. And proteins, these actually bind to integrin proteins, which transmit signals between the ECM and cytoskeleton to induce cellular responses in changes to the environment. And then these are also some other extracellular components. So cell junctions, there are plasmodesmata in plant cells, which perforate plant cell walls and allows cytosol and other materials to pass through. Tight junctions prevent leakages. So as you can see in this picture, there is no space that any materials can get through. Desmosomes help anchor the cells and form cell-to-cell attachments. And gap junctions are in animal cells, but are kind of similar to plasmodesmata in that they allow for cell content exchange. Okay. Let me see if there's anything I should answer. What is going on in the chat? Oh. Okay. Yeah. Anyways, next is Camel chapter 7, which is membrane structure and function. So, first, the lipid membrane is made of phospholipids, which are amphipathic, which means it has both hydrophobic and hydrophilic ends. So that means they will orient themselves with the hydrophilic heads on the outside and hydrophobic tails on the inside. And then this, along with proteins that are dispersed within the membrane, is called the fluid mosaic model. Phospholipids can shift easily within the membrane, and cholesterol, which we talked about as another type of lipid, helps maintain fluidity in cold temperatures. So membrane proteins, there are two types. One is integral proteins, which penetrate through the lipid bilayer. So these integral proteins often have both a hydrophilic portion and also a hydrophobic portion, and the hydrophobic portion is usually the part that is directly touching the phospholipids. Also, peripheral proteins are loosely bound to the bilayer, and they can be on either side of the membrane, which means they can be on either the extra or intracellular side. The intracellular proteins could be secured to the cytoskeleton on the inside, and the extracellular proteins are secured to the extracellular matrix on the outside. So many functions of these proteins include transport of ions or other substances as a channel or an active pump, signaling cell-to-cell recognition and enzymatic activity. So there are also glycolipids and glycoproteins, which are carbohydrates bound to lipids or proteins, and they help with cell identification. Phospholipid bilayers have selective permeability. Okay, which means they only allow small uncharged molecules through easily, since the inner part of the membrane is nonpolar, and substances such as O2 and CO2 are very small, and they are also nonpolar. So therefore, transport proteins are needed for large or uncharged molecules. So these channel proteins act as a charged or polar tunnel for these molecules, and if they are very big, they also have the same function. For example, aquaporins help transport water faster. Although water can diffuse through, it is very slow because of its polarity. And carrier proteins hold onto the molecules and changes shape for it to pass. So all in all, selective permeability is dependent on membrane structure and transport proteins. So there are two types of transport. One is passive transport, which is diffusion. So molecules tend to diffuse from areas of high concentration to areas of low concentration. And this movement is down the concentration gradient. So the molecules have a tendency to fill up more space. And this is passive because this does not require extra energy. And these molecules will diffuse down the concentration gradient if you just leave them there. And osmosis is the diffusion of water. So basically, water flows from a lower solute concentration to a higher solute concentration because it technically is a higher water concentration, and it's flowing to a lower water concentration to balance the two concentrations out, as you can see in the diagram here. So the tonicity of the environment often determines the direction of water flow. So in an isotonic environment, the concentrations of the intracellular environment and extracellular environments are equal, so there is no net water flow. However, in a hypertonic situation, the extracellular solute concentration is higher, and the water flows out. So in a regular animal cell, the cell will become shriveled, and in a plant cell, the cell wall will remain, but the cell membrane, the inner membrane, will pull away, and the cell will like start to shrink. And in a hypotonic environment, the intracellular solute concentration is more high, so there's a net water flow into the cell. And so in an animal cell, the cell will start to lyse and explode because there's too much water inside. And in a plant cell, this is called turgid, and it is a normal cell and the cell will not explode because there is a cell wall preventing that. Charged molecules have to be helped through the cell membrane, which is facilitated diffusion. So ion channels and other channel proteins will help with this. So it basically provides an ideal environment for these ions to pass through. And many of them are gated and open or close in response to a stimulus. For example, the potassium channels in neurons open in response to an electrical stimulus. So the other type of transport is active transport, and this is a movement of substances against the concentration gradient. This requires energy and ATP since you are moving things against their concentration gradient in the way that they do not want to go. So an example is the sodium potassium pump in neurons, and this helps maintain the membrane potential. And then another example is the proton pump against the mitochondrial membrane in cellular respiration, which we will talk about later. And then they can be coupled together in co-transport. So basically the transport protein can use the diffusion energy of one substance to actively transport another substance. And then another type of so then here I will talk about some bulk transport that can also happen. So exocytosis is the bulk expulsion of materials. So basically you have materials in a vesicle, and then that vesicle fuses with the cell membrane, and all of the stuff inside gets deposited out. And endocytosis is the bulk intake of materials. So for example, you have phagocytosis, which is the intake of food particles and pathogens. So the cell membrane basically wraps itself, forms a pocket, and then wraps itself around the food particle that it wants to take in, and then forms a vesicle like that. And then pinocytosis is the intake of really small molecules or minerals, which is like cell drinking, and phagocytosis is like cell eating. And then receptor-mediated endocytosis is where receptors on the cell surface bind to materials which are taken in specifically for that. Okay, I am done with my lecture. Next, how many times? Okay, we have a couple more minutes, so I have some usable questions that we can do. So it'd be nice if you guys can answer in the chat. These are related to the things we did today. Oh. Yes, we do answer in the chat. Okay. I'll give you a minute, I guess, for this, and then oh, you go pull it up. Oh, you can do that. Aren't you the one setting up the meeting. Yeah. Didn't you do this before? It's okay. You guys can just message me the answers. I'll give you till 7:55. Oh, okay. That works. Why are there 31 people here, but 16 of you guys answered? Actually, there's less, but you guys answer. Because this is taken from USIBO past exams, so okay. Anyways, I can end the poll right now. So most of you guys put E, which is the correct answer. And I can explain why. So I mean, I guess it is a type of lipid, which is, but it says the best answer. So unsaturated lipid is the best answer. So it is definitely not a carbohydrate because carbohydrates have carboxyl groups, which we don't see here. So that is not a carbohydrate, and it's usually in rings, in like less smaller rings. And it is a type of lipid, but we'll come back to that later. It's not a nucleic acid because it only has C, H, and O. And nucleic acids have a nitrogenous base and a phosphate group, which this is not, and this is also not a ribose. This is not a saturated lipid because it has double bonds. As I said, saturated lipids only have single bonds in their carbon chain. And so this is a saturated lipid. So it's kind of process of elimination, but it is a lipid since it has a very long CH chain. Yeah. Okay. Okay. So most of you put A, which is good because that is the answer. Since okay, we talked about how water would flow into areas of higher solute concentration. So if the extracellular conditions are very high in salt, then the intracellular water will try to flow outside of the cell. However, if the cells accumulate compatible solutes such as glycerol and sucrose inside their cell, the water from the outside the water from the inside would at least not flow out of the cell since the cell has a higher solute concentration. they can prevent the entrance of protons in their cytoplasm. Well, that doesn't really have anything to do with high salt concentrations. And then they have less porins or transport proteins. But well, okay, I can't. Okay. And then they have more saturated fatty acids or unsaturated fatty acids, but if there's salt, if the salt cannot flow in anyway because they are ions, and then the cell membranes are nonpolar, so that will not happen either. Okay. We'll just finish on this one. It was on the slides. Like it was stated on the slides. Okay. So most of you put D, which is very good because if you can remember on the slides on cytoskeleton, I literally put microtubules form cilia and flagella. So that's just something you have to know and memorize. Yeah, okay. It is 8 now, so I think we can end here. Cool. Okay. If we do guys have any other questions?